Recording in progress. Okay, so today we're going to talk about genetic 101 and learn just basics of how the genes work and so on. So our genome is consists of 23 pairs of chromosome, and 22 of them are exactly the same. And the 23rd one is different between women and men. And it's interesting that for some species, XY will be female and XX will be male. But for humans, it's XY will be male and XX will be female. So it's could be different for different species. So here on this picture, you can see how machinery of genes works. And you can see at the furthest left top, it's your cell with nucleus inside, and the chromosomes look like letter X. And if we start unwinding it, as we go further close to the middle, you can see that the DNA consists of different letters, and it's T, G, A, C, and they some of them are complementary to each other, like T and A and C and G, and each three of those letters next to each other will be translated into different molecule, and because of or different part of the protein, and as a result of that, you will end up with different structure of protein. And here we can see that molecular machine, that it's like a reading books, that if we think that our letters in a word, we can substitute one letter for another, then meaning of the word will change completely. So similar stuff occurs with our chromosomes. And if you can substitute one letter for another, instead of T, for example, we'll get G or C, then the machinery will read it different. And as a result, the protein structure will become different. And the ability of that protein to work in where it's supposed to work can be slightly different. So that's what actually is gene mutation. Gene mutation is error in genetic structure. What can happen is a substitution. So for example, you have letter wall, and you can see WALL, but if you substitute L for, I don't know, H, then you can make WAHL. It will sound same, but the meaning could be very different. So that's substitution. Then you could have insertion, where instead of, let's say, wall, you add, I don't know, add some additional letter, and then the meaning will be different. Deletion is when the letter is disappearing and then the word shortens, and as a result, that again changes the meaning of the word, or here is a structure of protein. And translocation is when part of the word completely changes position and can travel from one place to another and gets inserted into a different place. And it can be inserted backwards. And again, as a result, there will be a big change in the structure of protein or meaning of the word. So we have what we call phenotype and genotype. Genotype is a combination of our genes, and phenotype is how our genes translate into physical structure or physical features. For example, here we have LDL processing. The LDL is a bad cholesterol, and there are several genes that are responsible for that LDL cholesterol production. And as a result, we can have very high LDL, which we call familial hypercholesterolemia, because of different parts of gene that got mutated. has abnormality, and it could be either at the level of LDL receptor or at the level of actual LDL particle or at the level of different place called PCSK9. That's where one of our medication works. And as a result, we will have exactly same picture. Clinical picture will be super high cholesterol, premature coronary artery disease, and atherosclerosis in leg arteries, heart arteries, brain arteries. But the genetic mutation responsible for the clinical picture can happen in several different places. So this is part of when we have different genes, but they lead to exactly same phenotype. And this is example of clinical picture of familial hypercholesterolemia, when people have cholesterol deposit in their ligaments, and you can see here an Achilles ligament super thick. Occasionally we can see that deposit on hands and around the elbows and so on. Usually it will be when people have double copy of mutation, and therefore cholesterol will be around 500, 600, even 1,000, with LDL between 350 and 800 and 1,000. So those cases are super rare, maybe 1 in a million, if that. If person has one copy abnormal and another one is normal, that we call it heterozygous mutation, then they will have LDL above 190, but usually not more than 300, 350, and total cholesterol will be probably 300, 400. There still will be increased risk for coronary artery disease and strong family history of cholesterol, but people with heterozygous mutation usually wouldn't have this pronounced deposit of cholesterol in ligaments. They still can have deposit of cholesterol in the eye. We call it arcus sinalis, and that is we can find that in patients less than age of 40. However, if it's above age 40, it may be just because of a natural aging and deposit of cholesterol because of that. The picture on the lower right, you can see there is a yellow spot on top of eyelid. That's xanthelasmas. It's again cholesterol deposits, and they could be seen in heterozygous and homozygous, and they do get better as cholesterol level goes down. I definitely have seen that. So this is a different scenario. We have MTHFR gene, and it plays important role and in methylation. And methylation is one way how our body gets rid of toxins. And it plays a role in DNA and just getting rid of estrogen, getting rid of catecholamines, different other chemicals. And as a result, if person has mutation of MTHFR gene, they may end up with a very diverse variety of symptoms. So here it's kind of opposite of familial hypercholesterolemia, where you could have a lot of different genes leading to exactly the same phenotype. Here you could have one gene mutation that leads to a wide variety of clinical presentations. This particular picture here shows us how homocysteine gets converted into methionine or cystathione, and so on and so forth. And elevation of homocysteine is frequently hallmark of MTHFR gene mutation, because there is not enough of conversion of homocysteine into methionine. And you can see that vitamin B12 plays big role, and actual folate plays big role. And therefore, people with MTHFR gene mutation will require to have methylated version of B12 and methylated version of folic acid, not folic, but folate, tetrahydrofolate. So There are other places where things can get wrong. There are different mutations down the road in this picture, and that can lead to homocysteine elevation. And depending on that, if we look at full picture here, we may suggest other supplements like TMG, and which is trimethylglycine, and it's called betaine. Occasionally, we have to suggest to take SAMI as adenosyl methionine, and occasionally we have to suggest to take a glutathione as well. So what clinical presentation can occur? So first of all, how often do we see that MTHFR gene? There are two places in the gene where things that we can have mutations. Technically, it could be in many more places, but those are the ones which are clinically significant. And one place is called C677T, and another one is A1298C. If you have two genes of C677T, that we call homozygous, or if you have one C677T and another A1298C, then we call it compound heterozygous. So there is a fair amount of Americans who have a genetic mutation in one or the other or in combination. And you can see here that 10 to 15% of Caucasian population will have homozygous, and 25% of Hispanic descent, and 30 to 40 will have just heterozygous mutation in American population. With A1298C, you can see that heterozygous could be up to 56%, but between 11 and 14% are homozygous. And homozygous when you have two copies, one from mom, one from dad. And if you have two copies, it's always more pronounced. And if you have one copy, depending on gene, it could be dominant or not dominant. So for example, a little bit sidetracked to make understand better. So gene of brown eyes is dominant. So if you have one gene from mom or dad, brown eyes, another one blue from other parent, then more likely than not, you will end up with brown eyes or maybe green eyes, but never you will have blue eyes. In order to have blue eyes, you have to have two blue eye genes, because blue eye gene is not dominant. We call it recessive. So with MTHFR gene, it's kind of, it's more dominant than not. And therefore, if you have genetic mutation, it's still clinically significant, but not as significant as if it were homozygous. So it's kind of halfway dominant. And here are the outcomes. See here, which you can see in patients with MTHFR gene mutation. There could be blood clotting and cardiovascular events, including strokes, embolism, and heart attack. And usually higher homocysteine level, higher chance to having those effects. Plus, it can have effect on mood, depression, anxiety, bipolar, schizophrenia. It has big role in colon cancer, leukemia, chronic pain, fatigue, nerve pain, migraines, recurrent miscarriages, pregnancies with neural tube defects like spina bifida, anencephalopathy, and so on. So you can see how one gene plays such a huge role in our body and can be so diverse. And that's where it's important to recognize. Other part that I like talking about when we talk about genetics, it's detoxification pathways. Just to remind you, we have toxins that get to our body, or even our internal sources like breakdown products of hormones or neurotransmitters. They all go through phase 1 which usually takes place in the liver, and it makes fat-soluble chemicals into water-soluble chemicals. And in phase 2, we will make it again more water-soluble, I guess, and we'll get rid of them through different pathways. And you can notice there are different CYP genes, and CYP genes usually located in mitochondria. And you can see on the right in phase 1 what different ones are responsible for. So metabolism of estrogen, caffeine, Cymbalta, aflatoxins, coumarin, nicotine, Crestor, warfarin, and so on and so forth. So if you know your genetics, you can be aware of which of medications you want to use, smaller doses or higher doses, more frequent or less frequent, because it plays super important role in that, in the choice of different medications. Now we use knowledge of this phase 1 and phase 2 pathways in psychiatry. They frequently will order genetics for patients who haven't responded to regular treatment, because possibly that regular treatment wasn't good for that particular patient, because the medication was either metabolized too fast or wasn't metabolized enough, and maybe there was interaction, and so on and so forth. So my prediction that in 10, 20 years, we all will be genotyped, and we all will have our genetics stored somewhere in a central area where a machine will help us to understand which medications to take and what would be the doses that preferred. Right now, as far as we know, for example, with warfarin, that is blood thinner, some people may require just 1-2 milligrams, some people will require 5 milligrams, and some others between 10 and 20 milligrams, depending on your CYP2C9 gene mutation. And that's clinically very significant. Similarly as for Plavix and proton pump inhibitors, because like there is a strong interaction between proton pump inhibitors and Plavix, especially if you have slow or high metabolism through this CYP2C19 gene. So as I said, there are different ways to look for those genetics, and let's talk about that. So genetic testing, you could use actual labs that are covered by insurance, or you can use other ways. For example, I use 23andMe, and I will ask for raw data and put it through Promethease or other pathways or other companies that can spit out the information about your detox pathway, for example. With geneticgenie.org, it gives you detox and methylation if you have Ancestry or 23andMe. And you can know for sure which of the genetic mutation in that detox pathway you have or you don't have. Of course, you have to then do the legwork and know what medication process through which gene and what to do about that. If that's a lot of work, and physicians usually wouldn't remember that anyway. So that's where companies like Alpha Genomics becomes very simple, because they will give you actual list of medication that are good for you or safe for you to use, or you should use with caution, or you shouldn't use because of the presence of genes. And they do that for psych medication, pain medications, and cardiovascular medications. Unfortunately, right now, I try to order or get the account with them again, and they're too busy with COVID testing, and they are not accepting new accounts. In any case, after you've gotten your results, you can order a genetic consult. In Cincinnati, genetic consult is available through Children's Hospital, and there is apparently, there are a few other independent geneticists who you can ask for consultation and take your data and discuss all the results and understand better how that translates. Again, you do want to remember that genetics load the gun, but the environment pulls the trigger. And for example, for same MTHFR gene mutation, if you have enough of methylated B12 and folic acid, it's not likely to create a problem. And if you have, on the other hand, in other genes, like if you have brown eye gene, you will end up with brown eye gene. There is nothing you can do about that. But still, there are tons of genes where you can do something because environment does pulls the trigger, and you can affect that environment. You can get the detox, you can get the proper nutrition, proper stress management, and so on. So let me